Good afternoon, ladies and gentlemen. On behalf of the Palaeobotanical Society, Lucknow, as a secretary of the society, I welcome you all in the fourth Mrs. Savitri Sahni Memorial Lecture, delivered by Dr. David B. Lazarus of Museum of Natural History, Berlin, Germany, on the topic Plankton and Climate Change. I am indeed very happy that Dr. Rajiv Nigam, Emeritus Scientist CSIR, has agreed to become the chief guest on this occasion, and Dr. Vandana Prasad, Director BSIP Lucknow, has agreed to become the guest of honor on this occasion. The first Mrs. Savitri Sahni Memorial Lecture was delivered by Dr. D.K. Upreti of NBRI Lucknow, which is known as CSIR NBRI or National Botanical Research Institute, Lucknow. And he delivered a lecture on the lichens. The second was delivered by Professor Subir Bera of Paleobotany and Paleontology Section, University of Calcutta, Kolkata. And the third one was delivered last year by Professor Achim Browning of Germany, and he spoke on the tree ring dating and climate change. And this year, the lecture will be delivered by a famous micropaleontologist and a renowned micropaleontologist who has contributed a lot on the plankton. And he will deliver the talk, Plankton and Climate Change. So without wasting our valuable time, I would like to invite Dr. David V. Lazarus to deliver his lecture. Dr. Lazarus,
yeah,
please.
it's okay.
All right, so this, okay, all right, so I'm going to get started here. This is indeed a talk on plankton and climate change. And I'm going to mention some studies from the biologist study of plankton, but my focus is really on the microfossil record, as I think it's particularly important, and of course that's my background. First of all, what is plankton and why is it even important? And that's, well, it's basically any organisms that live in the water column and their ability to move on their own power is so weak that they are largely instead moved around by the currents and water movements externally. These are mostly very small organisms. Many of them are protists, although there are also metazoans in the plankton. This is the largest environment on the Earth, and we don't know very much about it. It's a very remote environment, and we have a hard time studying it. But it, of course, is crucially important in many ways. It's the base of the marine food chain, and it's also important in cycling various elements in the Earth's geochemical cycling system, and particularly that of carbon, which plays, of course, a critical role in the form of carbon dioxide in the atmosphere and regulating climate. And how this interaction between plankton and the carbon system in particular are also of great importance, in addition to understanding the effect of the biota itself. And a wide range of size classes, going from bacteria, the so-called picoplankton, up through the microplankton, which is mostly protists, through and then eventually, of course, up to the top. I'm going to refer to the water, the ocean, the thing here on the right here. 50 meters or in the ocean, that's usually fairly warm. And then there's a transition zone, thermocline, and which is actually quite cold. And I'm not going to be talking about coastal environments today, but only about the epipelagic open ocean environmental situation. It's also important to understand that there is a biogeography to plankton, and we have polar biotas, and we have a broad cosmopolitan mid to low latitude biota, which is in yellow, and then we have forms restricted to the tropics. And we also have upwelling zones near the continents here in light blue. And it's important to remember that the polar biotas and the upwelling regions are the ones primarily responsible for the export of carbon into the deep ocean or into the sediments. Carbon export in the red and yellow zones is fairly minor, actually, although they're very productive. Most of the material is recycled in the upper water column. And this is an illustration of these various mechanisms by which the plankton exports and sequesters carbon from the atmosphere. And the phytoplankton export it both in the forms of carbonate and as organic material. A lot of this is remineralized by the zooplankton and by the bacteria, and then eventually upwelled, and some of the gases back out to the atmosphere. And it's also important to realize that when phytoplankton forms carbonate, it exports carbonate into the sediment, but at the same time, carbon dioxide is released into the atmosphere. So on short time scales of less than a few thousand years, it's only the non-carbonate plankton that actually has a positive effect on the sequestration of organic material and removal of carbon dioxide from the atmosphere. And so it's the interaction between these different groups as well as the overall productivity of the ocean that's important. And for some reason, there we go. Okay, good. Biologists have studied plankton for a long time, and in the 19th century, they focused on species descriptions and classical things like that, and then how distributions of higher taxa, such as diatoms as a group, and how that interacts with ocean geochemistry. And they've been adding a variety of new tools beginning in the 1970s. The distribution of plankton became known by using satellites to map the color. And in the last few years, environmental DNA surveys have been used to understand how many different species and higher taxa are out there and where they are and how abundant. And this is revolutionizing our understanding of the plankton, but this molecular data is so new that for the most part, it's not yet been integrated into the models of how plankton is going to respond to climate change. So whatever I say today is still very provisional and is going to change quite a bit in the next few years. And one of the things that's happened as a result of this new data is we're getting a better understanding of the complexity of the ecosystems. The early ideas about the planktonic realm were very simple. We had phytoplankton, diatoms, and coccolithophores, which were eaten by the zooplankton, copepods in particular, and then by a variety of fish. This view is partially due to the lack of information about the smaller organisms, but also partially because the agencies that funded most of this research were marine fisheries organizations, and they were mostly interested in the fish side of the cycle. Okay. Having a slight problem getting things to move here. Okay. And we know nowadays that the plankton has a much more complex food web. We have the phytoplankton and we have the mesozooplankton, the copepods, but this microzooplankton, the protists play a major role in mediating between the two. The bacteria play a major role of recycling elements in the water column. And so it's a much more complex system than we knew about in the past. And climate change studies in biology both are at these higher level taxa levels with geochemistry, and I'll get onto those in a moment. But there are also studies of how individual organisms respond. And there isn't a lot of species level data. As I noted, they've stopped doing a lot of the species level studies, but there are a couple data sets of critical importance, the most important being the Plankton Recorder Project, which in the last 50 years or so has routinely, regularly recorded the abundances of a select number of species, mostly from the North Atlantic, using very simple mechanical devices as shown here. And these types of studies have shown a pattern of migration and changing seasonality, even in the plankton over the last 50 years. And though there have been cooling as well as warming episodes, just look at the warming episode data for a series of species listed here, both phytoplankton and the copepods. The colored bars here show magnitudes of northward change, mostly northwards, of 200, 300, or even 500 kilometers. So we know that there's already a lot of change going on in the plankton today of migrating in response to climate change, but also that these species are not migrating at the same rate. And therefore, at any given location, we're starting to see the development of new communities with different predators and prey species in association than existed before. And there's also changes in the phenology, that is the seasonality of the organisms, of up to 1 month. This is a plot of the change in seasonality of different species. and scaled by months. And you can see some of the dots are that high already. So there's already a lot of change going on in the plankton. Excuse me. And the modeling studies based on the higher level taxa are also looking farther into the future. Here are a couple examples. On the left-hand side is an estimate of what the Southern Ocean biomass might look like for the phytoplankton 100 years into the future. And it's a complex pattern of response. In this model, some areas of the ocean become significantly more biomass, and other areas have significantly less biomass. Studies have also looked at how climate change might affect the distribution of diversity. This is an example using as a baseline one model, climate model prediction of temperature change, and the estimated change in the local diversity as a result in the lower panel, with changes of 30 or 40% possible, particularly higher diversity in polar regions. So, excuse me. The same types of studies can be used to understand how some of these important taxa, such as the diatoms, might change and thus affect the ability to export carbon in the system. Here's a model that shows major drops in the relative abundance of diatoms in the ocean, up to 50%, although that's mostly in polar regions. Lower latitudes don't show a lot of net change. But nonetheless, 15% or so decline in the export productivity of diatoms in the future with high levels of PCO2, which are similar to some of the worst case scenarios for projected future global warming. Okay, the biologic studies, to summarize them, show that shifts are already occurring, and they're fairly substantial, and larger shifts are predicted for the next 100 years. The response, however, is going to be complicated, and the overall impact on the productivity and carbon capture ability of the oceans is not entirely clear, but thought to be fairly modest so far. But these models always assume that the elements in the predictions, which are in the equations, such as diatoms, are behaving in a constant way. There are simply constant parameters in the system. And if we, however, have changes in the composition of those diatom assemblages, or in the worst case, if some species became so rare or even went extinct that they no longer effectively were part of the system, then these models are no longer accurate and they can't predict what will then happen. But understanding how such things as the actual extinction of species might occur is very difficult in biology, because almost by definition, species that are still alive are not necessarily good subjects for the study of extinction. So traditionally, you go to the fossil record to understand extinction. And we do have a fossil record for the plankton. We don't have a lot of groups that fossilize, but we do have both phytoplankton and zooplankton groups. We understand to a much better degree than for other types of fossil records how the plankton has fossilized and how good our record is. And it is very complete for certain groups. And we have huge numbers of specimens. I've estimated that we only have 10 to the 15th specimens so far that we've recovered and can actually study, which still is nonetheless a huge number and vastly more than all of the types of fossils put together. And there are different types of plankton that preserve. We have the phytoplankton groups, including the coccolithophores and diatoms, with their different chemistries of shells, and there are different impacts on the carbon system. We also have zooplankton, which are here mostly listed as mixotrophs, because we know that for the microzooplankton, these protists, most of the zooplankton groups also have photosynthetic symbionts, and so they actually use both modes of nutrition. And we know that at least for some groups, excuse me, such as the radiolarians or the foraminifera, the percent of the species preserved is actually very, very high. So we have very complete records to work with. And here are 3 different time scales of response of plankton to climate change in such studies: glacial, interglacial scale studies of the last few glacial cycles, studies of much more ancient events, sometimes very dramatic events that occurred over a space of anything from 100,000 years to 1 million years, and then the overall record, not just picking certain subsegments, but looking at the whole time change of series over periods of tens of millions of years up to 100 million years. So here's a glacial interglacial type study. And in these studies, starting with CLIMAP, and the current version of these types of studies is called Margo, the primary goal is to reconstruct temperature change. And the fossils are used primarily as proxies for temperature. So they report only net temperature results rather than the behavior of individual species and the data sets that they've collected. But this at least is also interesting, and it points out this general phenomenon of climate change, that the climate changes much more dramatically in polar regions. This is the last glacial reconstructed. Here, temperature changes are up to 10 degrees centigrade in the oceans, and very little change is seen in the tropics by comparison. And if you go into the actual data sets that these studies are based on, you can see what happens to the individual species. This is just a very small fraction of the available data showing a change in that. These are different species of radiolarians, and over the last 12,000 years, and you can see indeed very complex and very individualistic patterns of response to changing ocean conditions, which is similar to what the plankton recorder studies were showing on a time scale of decades, that you get changes in relative composition at any given time because the species are responding individually. And though I don't show it here, we also know that one glacial cycle has an assemblage similar to the previous glacial cycle or interglacial. And so the composition of the assemblages is very resilient. And in the last few glacial, interglacial cycles, we have not seen much in the way of either evolution of new species or extinction of species. So one gets the impression that the primary response is going to be one of changing relative abundances and relative locations, but nothing much more dramatic than that, which is sort of nice. It's coherent with the biology studies. And we also know cycle. Here is from an equatorial Pacific study. There are many such studies. Change in the export of organic carbon into the sediments. This is the last maximum here. And this is accompanied by indeed much lower concentrations of carbon dioxide in the atmosphere. This is the green line. And this is directly from ice core studies where they actually have the, bubbles of air from the past to measure the concentration in. And this was probably ultimately controlled by the availability of nutrients into the system. Here, input of trace elements that are essential for the plankton, such as iron, through dust from winds, where you can see a peak in the glacial and a decline into the interglacial. However, there's a major problem with all this, and that is that the average length of a species, the evolutionary longevity, that's on the order of a few million years. And so our living plankton has experienced not one or two or three, but dozens of really major glacial interglacial cycles. And the species have adapted their biology to this range of environments, because although they can move and migrate to adjust their preferred temperature, all sorts of other things, such as the composition of the communities that they're in, or the irradiance that they see at a given latitude, or the seasonality, are to evolve and adapt to this. And their adaptive range is primarily to colder environments, not to the current environment. And they've been finely tuned to be survivors of this type of evolutionary system. But what we're going to be doing is pushing this environment way off the scale of the illustration into warmer conditions, completely unprecedented over the last several million years of evolutionary adaptation. And what will happen to these taxa when they are exposed to something like this? And here you have to go beyond the current glacial, interglacial tuned assemblages and take a look at what's happened in other intervals in the past. And one way to do this is to look at really dramatic changes that occurred in the past. And of course, the one that's the most famous and is best studied for the plankton is the Cretaceous Tertiary boundary, where we had this meteorite impact and we killed off some of the charismatic megafauna like dinosaurs, and it had a major effect also on the plankton. And we know from simulations and other types of data that there was a dramatic environmental change, climate change very rapidly. In polar regions, the temperatures dropped by 10 degrees or more in the space of just a few years. For several years, the solar radiation basically stopped. Here are 3 time intervals, second, third, and fourth year post-impact. And this is a log scale. So here you're looking at a fraction of 1% of the solar irradiance 2 years after the impact. So we had these massive changes in the environment. And we know that they had an equally dramatic change on the plankton. It occurred very rapidly. Here are the planktonic foraminifera records. This is from a deep sea sediment core. This is the impact layer itself. And these are just centimeters above and below. And here's the latest Cretaceous planktonic foraminifera fauna, all sorts of species. And then just centimeters above, we've lost them all. There's only a couple small surviving species that are still present. And we know that this happened very rapidly in immediate association with this impact event. And you can see it in many groups, although not all groups, of plankton. If you ignore this yellow part, which are something other than actual coccolithophores, there are about 90 some species of coccolithophores, the calcareous phytoplankton in the late Cretaceous, and they basically all went extinct except 10 of these species. And over a period of several million years, we had the evolution of a new assemblage of Cenozoic forms. And we know that such a massive extinction event in the plankton has a massive effect also on the ability of the ocean to export carbon. Here are multiple proxies for the ability of the ocean system to capture and sequester carbon. And you can see, for example, the accumulation rate of carbonate in deep sea sediments dropped dramatically at the KT boundary and stayed low for nearly 2 to 4 million years afterwards before it finally recovered. So extinctions have a huge impact also, when they're large enough, on the ability of the ocean to function as a carbon capture system. But the KT boundary is much more dramatic, we hope, than anything that's going to happen to us in the future due to global climate change. We don't have global darkness and so forth and so on. So it's not necessarily a great analog. It gives us an idea of the scale of response that's possible, but we need to look for something a little bit more moderate if we want to understand what may be in store for us for our future. And this is where these long time-term studies really come in. And you can just use the catalogs of the recorded first and last appearance of different species as one database. This is an example here. These in individual samples, and that we use for that the Naturkunde database that we developed and which we run in Berlin. And this gives you a chance to understand, quote unquote, more normal behavior of plankton in response to climate change. And here are a couple examples that I'm going to show here. And they're mostly from our own work. And I'm going to skip over some of the others that are out there because there's just not time to cover them all. This particular study shows the diversity history of the diatoms in blue over the last 60 million years, and the history of the climate system, the mean state of climate based on the very often used and we assume that higher diversity of diatoms also means higher diversity or abundance of diatoms in sediments, and we have also data sets that show that. But what, and if you take out just the time variable and plot just the two variables here, you can see indeed that climate state and diatom diversity are very highly correlated to each other. A remarkably good correlation in paleobiology. And what's important here is that if we do go to 1,000 ppm atmospheric CO2 concentrations, which is what some of these scenarios show, we would expect a climate system similar to what last existed on the planet in the Eocene, which are shown by these red dots here. And at this time interval, only about half as many species of diatoms existed in the plankton. If temperature, state of the ocean system, and diatom diversity are linked, as seems to be the case here, then you would expect half the species that currently are alive to disappear, to go extinct. And it would be, of course, those species that are most closely adapted to cold environments. As things get warm, the ones that are further away from the new optimum are likely to be the ones that go extinct. And this is a plot showing the origination times of the living diatom flora. And the polar species are in blue and purple. The tropical ones are in red. And you can see that most of the living polar plankton species originated only in the last few million years, as the Earth's climate had cooled to the point that northern hemisphere glaciation was starting somewhere around 5 to 3 million years ago. Indeed, some of the key export species today, shown here by black triangles, are typical cold water species. And so it would be the loss of species in polar regions, which, as I said, is where nowadays the primary export of carbon is occurring, that would be most likely where we'd see extinction of species. This is based on this one study. And there also is evidence from the same type of data sets that as diversity changes in the diatoms, you also see changes in the atmospheric concentration of CO2. This red curve is one estimate of atmospheric concentration over the last 20 million years of carbon dioxide, shown here. And this is the diatom diversity data that I showed earlier. And you can see there's a very strong correlation between the two. So we see also on this scale the link between diatom diversity in the plankton and the ability of the ocean to sequester carbon and thus affect atmospheric PCO2 and climate. Now, the last study I want to show is to go back to this question of extinction. And this is this most recent paper that Sarah Trubowitz, working with us, published in Nature Communications just a few weeks ago. And what Sarah did for her thesis was to try to estimate the diversity of radiolarians, which are this important group of microzooplankton that helps recycle carbon between the phytoplankton and the copepods and so forth, and looked at the diversity of this group in the tropics over the last 10 million years. And this is her best estimate of the diversity there. And what she found was that the diversity didn't change very much. But on the other hand, neither did the environment. This is a compilation she made of the temperature change in the tropics over the last 10 million years. And there was only about 3 degrees of temperature change. So, okay, this is perhaps not a terribly exciting result by itself, but she went further and then compared this to what happened in the polar regions. And this is based on radiolarian diversity data over the same 10 million year interval, but now for the polar regions, particularly the Southern Ocean. And this was done by Joanna Nodi, the postdoc working with us in Berlin in his thesis a few years ago. And the diversity in the Southern Ocean originally was fairly constant. And then although it stayed constant, you started to see changes in the evenness structure, the ecology of the radiolarian biotas. And then around 6 million years ago or so, there's a bit of a gap in our data set, you start to see a massive increase in the extinction rate. And you start to lose lots of species. And then although the extinction rate doesn't go back down to the background level before, it does drop. And eventually you reach the modern condition where you've already lost half the species. You go from around 200 to around 100 species in this time interval in the polar regions. And this corresponds to a temperature change in the polar regions of nearly 10 degrees. This is a compilation of the temperature change history in the poles that Sarah put together for this. And so you have a loss of nearly 50% of the radiolarians in response to nearly 10 degrees of temperature change, which is a scale response very similar to the 50% loss of diatoms to a 10-degree change in temperatures between today and the Eocene that I showed in that diatom study earlier. And what Sarah was also able to show, which was not possible in the earlier study by Joanne, is that species did not respond primarily by migration to the temperature change. Although this was a cooling event, and therefore somewhat warmer adapted species in the poles should, in theory, according to the biologist's model of migration as response, they should have simply migrated somewhat equatorward. This is largely not the case. This is numbers of species going extinct in each time interval, these bars, and the ones in green are the polar species that indeed migrated equatorwards. The blue species are cosmopolitan species that were present in both high and low latitudes and simply disappeared from the high latitudes. The red species are the ones that were endemic to the Southern Ocean and did not migrate and instead went extinct. And they make up the majority of the record. So there is a real disconnect between what the fossil record tells us and what the biology studies tell us about what's going to happen in response to climate change. Migration doesn't seem to scale up to the temperature change magnitudes that we're seeing in the fossil record. Instead, species go extinct. So to summarize the talk, the main points here are that studies of living plankton and Pleistocene marine microfossils show a similar pattern of response to climate change. We have latitudinal migration. We have the formation of new assemblages and communities, but overall resilience and little extinction. But the living plankton is a very special residual biota that's left after numerous cycles of glacial interglacial change, and is not necessarily a good example of what's going to happen to substantial warming in the future. And if you look at time series studies of how plankton responded to similar magnitudes of change in the past, when temperature changes reached 6 to 10 degrees in polar regions, the species didn't migrate. They simply went extinct, and substantial numbers of them. And thus, if this were to happen, this means that all these models we have as to how the plankton is going to respond to climate change and how plankton is going to regulate the global carbon cycle might very well be wrong. If we lost a lot of these carbon sequestering species, climate change would be worse, and the recovery in the far future would be hindered or simply impossible. So I believe that's just about it. I'd like to thank, first of all, everyone for listening and for the invitation once again, and also thank the people in my group and some of the external partners who've worked with me on doing this work. And I'm available now for any questions if we have any time. Okay, I can stop sharing my screen.
Thank you, Dave, for your exhaustive lecture on the plankton and climate change. And I hope that all the participants who has logged in today as a student or as a research scholar or as a postdoctoral fellow, as well as the scientists who are working in different parts of India, who are the members of the society. First of all, my thanks to all the members of the society as well as the scientists of the institute for logging in. And but the formal vote of thanks will be delivered by Dr. Ratan Kar. Dr. Ratan Kar. But before that, if participants have any question, they can ask. David, they can ask, and he will be very happy to receive your question. Or I have posted in the chat box that if you have any question, you can post it, and we will send by email to him so that you can get the proper answer. So if there is no question, I would like to request Dr. Ratan Kar to formally deliver the vote of thanks.
Am I audible?
Yeah, you are audible.
Okay, it's my pleasure to propose the formal vote of thanks on behalf of the President, the Vice President, Secretary, other office bearers, and members of the Palaeobotanical Society. I thank Dr. Dave Lazarus for delivering the 4th Mrs. Savitri Sahni Memorial Lecture on the topic Plankton and Climate Change. We are indeed delighted that he chose the very topical issue of climate change and how very important planktons are, or they play a major role in recording and predicting such changes. I'm sure that many of us here, especially the students, would have been greatly benefited by his lucid lecture. We're extremely thankful to Dr. Rajiv Nigam, who is well known to all of us here, and in fact, he's like a mentor here to all of us, to give his consent to be the chief guest. His presiding over the function is all the more relevant as he is one of the foremost experts on the said subject himself. So thank you, sir. On behalf of the society, I thank our director, Dr. Vandana Prasad, for being the guest of honor. I further pay our gratitude to her for providing all the logistic facilities for the smooth functioning of the Palaeobotanical Society, because the society runs in the premises of the institute, and without her kind support and help, the function of the society would not have been possible. I thank all the other office bearers of the society for making this function of success. I thank for participating. Thank you.